I'd like to thank members of the board for allowing me the opportunity to talk about some of the work that we've been focusing on, utilizing stem cells and evaluating their potential to treat multiple sclerosis. I'm going to begin actually at the end, and so as no doubt many of you are aware, the CIRM grants are often comprised of multiple investigators, and this is certainly the case with our recently approved ETA award. And I really would not be standing here without the great support and friendship of Dr. Jean Loring, who is an internationally recognized expert within the field of stem cell biology, as well as the director of the Center for Regenerative Medicine at Scripps Research Institute. Dr. Loring is the co-investigator on the ETA award and is intimately involved with all the data that I'll be presenting today. In addition, Dr. Claude Bernard, who is the director of the MS Research Laboratory at Monash University, is also involved in this grant that was approved for funding, and his area of expertise is in neuroinflammation and multiple sclerosis. Now, we're at the front, but the real people that are doing the work are highlighted right here, and these are members of my laboratory, Dr. Chen and Dr. Liang, who have done heroic work in the animal studies that I'll be showing you. In addition, Dr. Laurent at UCSD, Ron Coleman, who's a member of Dr. Loring's lab, and then Carmel O'Brien and Andrew Laslett in Australia have also provided extensive expertise in the work that we'll talk about today. And I thought I'd just give you a brief outline what I'd like to talk about, and I'm going to reassure you that I'm going to stay on time. I'm going to provide a brief overview of multiple sclerosis and just to orient you to what this disease is. I'd like to highlight some of the preclinical animal models of MS that we use to evaluate the therapeutic potential of stem cells, and then conclude with showing that human neuroprogenitor cells mediate sustained recovery in a very important and unique viral model of multiple sclerosis. And so anytime you talk about chronic disease of the central nervous system, I think it's important to introduce key players so that we can continue with our discussion. And so highlighted in this cartoon is a human brain, and early neuropathologists, when they looked at the human brain, would divide it down the midline and say this area is gray and this area is white. And that stuck. So there's the gray matter and the white matter. And within the gray matter, there are discrete populations of cells that populate this. And so, of course, there's the neuron that allows us to do what we're doing today. But in addition, there are glial cells, and these are astrocytes, which are the most prominent cell population within the central nervous system. These cells provide structural support as well as secrete factors that allow neurons to survive and replicate. And in addition, there's the microglial cell, which also secretes trophic factors for other cells of the CNS, but also provides important immune surveillance. Now, if you look in the white matter, and this is really where I want to jump off into the talk, the cell that we're very interested in with regards to MS is the oligodendrocyte. And this is a very important cell because it's the subject of attack by the immune system. And so what this cartoon highlights right here is the oligo. And so this cell produces myelin within the central nervous system. And the easiest idea to think about what myelin does, it allows for the conduction of nerve impulses from neurons. And so shown in this cartoon, you see one oligodendrocyte extending processes from the cell body. And these processes wrap around axons which extend from a neuron. And this cartoon, this exploded version, shows that there are numerous wraps of this myelin around this axon. And it's the presence of this myelin sheath that allows for motor skills that we all enjoy to occur. And this is because these nerve impulses will be transited down the axon. The easiest way to describe it in another way is that you have this electrical wire right here. This rubber sheath you could consider to be the myelin, protecting the electrical cord that's inside, allowing for the conduction of the electricity. And so this is important to consider when considering MS because this is a demyelinating disease, and so the myelin sheath is lost. So in simple terms, what is MS? It's an inflammatory disease of the brain, optic nerve, and spinal cord, and collectively you can refer to these as the central nervous system, or CNS. Any region of the CNS can be attacked in a patient with multiple sclerosis, and the symptoms really depend on the location of the inflammation and myelin destruction within the central nervous system. Importantly, it is the most common cause of non-traumatic disability in young adults in the United States and Europe. And so the steps that lead to the development of MS are highlighted in this cartoon. And so when you think about your immune system, you normally think about it as a friend, and you should, because your immune system is comprised of cells, and in particular T cells, which help orchestrate your defense following microbial infection. Without these cells, we would be in bad trouble. Now, in some individuals, though, this immune system can go awry, and instead of recognizing pathogenic microbes, which they certainly will do, this immune system will start recognizing your own tissue in your body as foreign. And this then leads to the development of what's known as an autoimmune disease. And in MS patients, the immune system starts recognizing the myelin sheath. And so activated T cells that are reactive to myelin are able to cross the blood-brain barrier and participate then in the destruction of the myelin sheath, which then impairs nerve signals that are transited down the axon. Another way to look at this can be shown here. Now, this is a white matter tract, and if you remember as I introduced the brain, it's the gray and the white matter. The white matter is where the myelin is, and the myelin is being produced by the oligodendrocytes. And so here, you can see axons which are highlighted in red, and this is a cross-section, and myelin is determined by the green that's encircling these axons. And then here is an MRI. We're looking down at the top of an individual that does not have MS, and these are the lateral ventricles, and so there are no lesions. In the center here is the vasculature, so this allows for the conduction of blood or allows blood flow. Now, in a person with MS, you see an active lesion, which is highlighted here in the MRI, shown here, here, and here. And what happens is these activated T cells, along with other cells known as macrophages, will infiltrate into the white matter tract and start engulfing and attacking the myelin. All right, and that's shown right here that these cells, these macrophages, are activated by T cells, and they start stripping the myelin sheath off of the axons. And so this is an active lesion. Now, over time, what can happen is you start seeing the number of inflammatory cells wane, and there are still a few that might percolate in. But importantly, the myelin is now completely stripped off these axons. And the problem with that is it impairs the conduction of nerve impulses, and more importantly, over time, these axons can undergo degeneration. And that's a very important aspect related to understanding the pathogenesis of multiple sclerosis. And so the natural history of MS can be highlighted in this slide. And so at the top, there's a very dramatic illustration over time of a patient that you see a dramatic reduction in brain volume. And this does not occur in every patient, but this can occur in many. And this is, you see the lateral ventricles enlarge, and you see numerous dropout of cells in the brain. And if you look down here, you see a progression of the disease. And the important points I want to highlight here are just during preclinical phase, when a patient doesn't realize he or she has MS, there still will be activity or demyelination initiating in the central nervous system of that individual. Clinical attacks will then present, and by far and away, the majority of patients with MS are diagnosed with what's known as relapsing, remitting forms of the disease. And over time, and this can be years, they will have periods of clinical attacks, and then these will subside. And there will be an interval of time where the disease is clinically silent. However, histologically, it's not. And then these arrows indicate MRI activity. Now, over this time, the interval between clinical attacks can shorten until eventually patients will enter what's known as the progressive phase of the disease. And this is a very important aspect related to MS because there are really no treatments for patients with progressive forms of multiple sclerosis. So who gets MS, or what causes MS? This is a very complicated question. It's relevant to not only MS, but many other autoimmune diseases. And what we know is there's a genetic predisposition, as well as environmental factors. And what I mean by that is for close to 100 years, investigators within the field have thought that there might be something like a virus or a microbial pathogen that can infect a patient that might have genetic predisposition. And this can lead to then autoimmunity, that is the attacking of the myelin sheath, which ultimately culminates in the loss of myelin and nerve fibers. So who gets multiple sclerosis? As I indicated earlier, it's generally a disease that's diagnosed in young people, usually between the ages of 20 and 40. About 400,000 people within the United States are diagnosed with MS, And again, similar to many autoimmune diseases, women by far and away have a much higher prevalence of developing multiple sclerosis compared to men. And so within California, there are an estimated 33,000 patients that are registered with MS. And if you think about this, then there's over 200,000 individuals affected by the disease. And what that means is caregivers, family members, employees, employers that are all affected by multiple sclerosis. And so when Dr. Loring and Dr. Bernard and I sat down to think about how we wanted to approach this disease with regards to stem cell treatments, we thought, well, in the beginning, neuroinflammation, that is, the infiltration of immune cells into the central nervous system, is critical in establishing the disease and driving the pathogenesis. But over time, neuroinflammation wanes, and it's really neurodegeneration that's critical in driving really many of the clinical aspects related to the disease. The majority of drugs that are available to MS patients focus only on this aspect, immune modulation, that is, how can you dampen immune cell infiltration into the central nervous system. And what's absent in terms of a drug or drugs that are available to patients with the progressive form of the disease are drugs that can promote or attack or prevent neurodegeneration. And so we became very interested in cell replacement therapies in terms of trying to not only impede the progression of the disease, but promote repair of the damaged nerve tissue, specifically remyelination. And so the goals then of our grant are to promote repair of the damaged nerve tissue and improve motor skills, limit the progression of ongoing disease, and importantly, no dangerous side effects that we can address or see. And so the model that we have utilized is a very aggressive form of MS. It highlights key aspects related to the relapsing remitting form of the disease, but importantly, it also encompasses aspects of the progressive form of the disease. And so we've used a neurotropic virus to infect animals, and then these animals, and we do this by an injection into the brain, these animals over a short period of time will develop clinical symptoms very similar to what you see in a patient with MS that correlates with the inflammation within the central nervous system. And so here are mice 3 weeks post-infection with our neurotropic virus. And you can see they have a very severe paralysis. They cannot right themselves. And if you look in the spinal cords of these animals, and this is representative examples are shown here, this is a control or sham spinal cord that's been stained with a dye that allows for visualization of the myelin sheath, which is highlighted in blue. All right, so this is a cross-section of the spinal cord. The gray matter is kind of more purple. And what's important to notice here is the myelin sheath is intact, looks really good. In contrast, in these animals with paralysis, you can clearly see loss of this blue staining, and that's indicative of demyelination. If this is just another way to visualize this, it's a toluidine blue stain that allows us to look at axons, and I can't see it from here because there's this light shining in my eye, but you can see that myelin is being stripped away from these axons and compared to the control animal. And then this is what's known as a sagittal section of the spinal cord, or in other words, the length. And this shows these great swaths of myelin damage that occur that's correlative with then immune cell infiltration. And that's why these animals are undergoing paralysis. And so the question we had is, does transplantation of human stem cell-derived neural precursors, or hNPCs, into mice that are persistently infected with a neurotropic virus influence the outcome? And the rationale here is very straightforward. Viruses have long been considered important in initiating demyelination in humans, and equally important is that numerous neurotropic viruses exist that persist within our central nervous system. So we concluded that it was important to evaluate the therapeutic potential of human neural precursor cells or progenitor cells within the presence of a persistent viral infection and that correlates with chronic neuroinflammation and demyelination. And so what are the cells that we're working with? These are images provided by Ron Coleman, who is in Dr. Loring's lab at the Scripps Research Institute. These are human embryonic stem cell-derived neural progenitor cells. Here is just a stain for the nucleus, and these neural progenitors will express PAX6. You can combine these images, and the purpose of this is just to show that we have a very enriched population of neural progenitor cells. And these are the cells then that we are going to put into those animals with paralysis, as I showed you earlier. So what happens when we do that? If you look at panel A right here, we injected these cells in a very specific manner. We did an intraspinal injection, and I can go into that in more detail if you have questions after the talk. But what happens when we take paralyzed mice, and on the y-axis here is the severity of clinical disease, that is the motor skills, 0 being nothing, the highest point 3.5 or 4 being severe paralysis, and we transplant in the human neuroprogenitor cells, we see a marked improvement in motor skills in these animals, in contrast to mice that receive either human fibroblasts or vehicle alone. If you expand this out to 6 months post-transplant, you can see in the red that the clinical improvement that we observe in animals transplanted with our neuroprogenitor cells is sustained. And that's really remarkable compared to control animals. We injected 250,000 human neuroprogenitor cells. We have concluded that the recovery is dependent upon the number of cells injected. Furthermore, if we do either an intravenous or intraperitoneal injection of the cells, it does not result in improved disease outcomes. So therefore, the route of administration is critical. So what does this look like? So before I play these videos, these are 4 mice that have established demyelination and inflammation within the CNS that are paralyzed. And then what I'm going to show you next is the same mice 3 weeks post-transplant with human neuroprogenitor cells. So similar to the movies that I showed earlier, you can see that these animals have very serious paralysis. This is prior to transplantation. This is actually the day prior to transplantation. These same animals then, following transplantation, we see a dramatic improvement in clinical recovery in these mice. And so we have done over 100 injections in animals, or over 100 mice have been injected with these human neuroprogenitor cells. And this image, or this movie, really captures what we see. About 76 to 80% of the animals recover motor skills. And by recovery, we mean by a dramatic restoration of the ability to walk and stand. Now, very importantly, is that if we go out to 6 months post-transplant, are these motor skills retained? Or is this recovery retained? So here are representative mice, 6 months with the disease for 6 months. These are animals that have been transplanted with the human neuroprogenitor cells 6 months later, and you can see now they have a little bit of impairment, but they have sustained ability to walk. So this is obviously very exciting. When my postdoc presented me with these data, I didn't believe her, and I said, I mean, the mice are all coded right, and I had to go down and score the mice myself, and so I still, anyway, it was very exciting. So what happens to these cells? Well, importantly, and this is actually a point that Dr. Tashari brought up when you guys were discussing the inflammatory bowel disease grant. In our mice, these are immunocompetent animals. Otherwise, they wouldn't get the disease because it's an immunologically mediated disease. Importantly, when we transplant the cells, we are not administering drugs that modulate the immune system. They're entirely immunocompetent. So in other words, those cells that we put in could be recognized as foreign and rejected. And so how we looked at this was Ron Coleman in Dr. Loring's lab engineered these cells to express luciferase. So the human neuroprogenitor cells now express luciferase, which is an enzyme that's actually cloned from fireflies that allow them to glow. And we can actually visualize these cells at defined times post-transplant in the spinal cord. And you can look here. You can see as early as day 1, we can clearly see the signal of the cells in the spinal cord, but over time, you see this gradual diminution in the level of signal. So the cells are being rejected or disappearing below the level of detection. So clearly they're going away, or, in my opinion, they are being rejected. But why then are the animals improving with regards to their motor skills? And so what we found is fairly remarkable. Shown here in the top panel are clinical disease graphs for an individual representative mouse transplanted with media. And this is the time of transplantation, and this animal remains paralyzed. In contrast, an animal transplanted with the neuroprogenitor cells, we see, as I indicated in the other graphs and movies, improvement in motor skills. We have taken spinal cords from these animals and looked at the severity of neuroinflammation. And in particular, we're looking at macrophages and microglia, which participate in myelin destruction, as well as T cells. And what you can appreciate, the brown here in the white matter tracts, which are outlined in the red hash marks, are filled with macrophages. And so that's the cell that's contributing to demyelination. Higher magnification shows also numerous inflammatory T cells present within these animals. However, in animals that have been treated with the neuroprogenitor cells, after 3 weeks, what you see is remarkable. A dramatic reduction in the severity of neuroinflammation is characterized by reduced macrophage infiltration as well as T-cell infiltration. So somehow these cells are dampening the ability of immune cells in the periphery to gain access into the central nervous system and more specifically access the white matter tracts. In addition, what we've determined is that these cells also modulate immune responses by T cells. And so not to belabor this point, but we've done very elegant ways to measure T cell responses in animals treated with these human neural progenitor cells. And what we've concluded is that we see a dampened production of a pro-inflammatory cytokine called interferon gamma that exacerbates inflammation, and then elevated expression of an anti-inflammatory cytokine called IL-10. So in other words, following human neural progenitor cell transplantation, this improved clinical disease correlates with muted neuroinflammation and altered T-cell responses that may direct this neuroinflammation. Now, what also is very impressive and dramatic is shown here. Now, if you recall, this is a coronal section of a spinal cord from a control mouse and a neural progenitor mouse, and now stained with a dye that allows us to visualize myelin. Now notice, this is 3 weeks post-transplant with either media or our neural progenitor cells. In the control animals that have severe paralysis, we see numerous areas of demyelination highlighted in red. These are the lateral white matter columns. This is the ventral funiculus, the dorsal funiculus. All have severe demyelination. In contrast, the human neural progenitor cell treated animals, we see only a mild bit of demyelination here, which we would expect because remember, these animals were paralyzed at the time of transplantation. You're not going to go to your physician and say, I might get MS. Can you treat me with a stem cell? You're going to have the damage already present, and that's very important to highlight that these are therapeutic applications of cells. What's interesting, if you recall, in the areas of demyelination, numerous macrophages and T cells contributing to this. We don't see this in the neural progenitor cell treated animals. It's a significant reduction in the level of demyelination. More importantly, in the animals treated with the human neural progenitor cells, we looked in the areas of myelin damage or loss of myelin, and what we observed is very provocative, and that is increased remyelination, that is the production of new myelin in the areas in which there was demyelination. And this is an extremely important point that I need to emphasize because this shows a therapeutic potential to regenerate myelin, and that's correlative then with the clinical recovery. So how are these cells doing that? Well, we have an interesting preliminary data. If we culture these human neural progenitor cells in the lab and then take the media that they're bathed in and then put this on top of mouse neural progenitor cells, the rationale being that something is being secreted by these neural progenitor cells that modulate the microenvironment that A, dampen neuroinflammation, and B, promote remyelination. Maybe they're doing something to local endogenous cells, neural progenitor cells, that allow them to mature and differentiate into myelin-competent cells. And what we found is if we take mouse neural progenitor cells and culture them with conditioned media from neural progenitor cells, what we see is enhanced differentiation into oligodendrocytes, which are, again, are the myelin-producing cell. So these neural progenitor cells are quite remarkable in their ability to promote repair. So to put everything I said into perspective, and I think I'm on time, intraspinal transplantation of human pluripotent stem cell-derived neural precursors into a mouse model of MS results in a sustained improvement in motor skills. And I need to, again, emphasize the importance of this. It's sustained. By far and away, the majority of preclinical models of MS that are employed, they look at very short time windows. And we really wanted to get at this, and we allowed the animals to go for a long period of time. The recovery that is mediated by the neural progenitor cells is associated with dampened infiltration of immune cells, specifically T cells and macrophages. Equally, if not more importantly, T cell responses are altered following transplantation. I showed you some data with regards to the secretion of cytokines. What I didn't show you is that pathogenic or disease-causing T cells are impaired in their proliferation following transplantation of these progenitor cells. The severity of demyelination is reduced in these animals, and more importantly, it does not spread. And most importantly, we see an increased amount of remyelination, so repair that's occurring. And we believe that something that is being secreted by these neuroprogenitor cells is mediating this effect. Because think about it, those cells are there for an extraordinarily short period of time, yet this recovery is sustained for at least 6 months. This highlights the ability of these cells to manipulate the microenvironment again and create an environment that's conducive for repair. So the directions that we want to take this, we want to test the efficacy of these neuroprogenitor cells in other animal models of demyelination. This is where our friend and collaborator, Claude Bernard at Monash University, is going to come in. He's going to use another very well accepted model of MS and evaluate the therapeutic potential. We want to identify the factors that are secreted by these neuroprogenitor cells that mediate the immunomodulatory effects, as well as enhance the differentiation of neuroprogenitor cells into oligodendroglia. We also want to define the mechanisms by which these hNPCs promote recovery and evaluate mechanisms associated with rejection of the human neuroprogenitor cells, with the idea being if they are so therapeutic in such a short period of time that they're present, if we can sustain them within the CNS, perhaps this is beneficial to the animal and to eventually humans. I'd just like to acknowledge my laboratory at UC Irvine. Dr. Loring, again, this work would not have been possible without her advice and guidance, and again, she's the co-investigator on the award. Dr. Loring could just as well be up here doing this presentation as I. Members of her laboratory, which have provided the cells that we're working with, Claude Bernard, who is, if you don't know him, he's just a wonderful scientist and a gentleman and a great collaborator and international recognized expert in MS. And then our other colleagues at CSIRO in Australia. And of course, I have been lucky enough to be funded by the California Institute for Regenerative Medicine for quite some time, and I would certainly like to take this opportunity to thank you for allowing us to go forward on our work on MS. So thank you very much for your attention. If you have any questions, I'd be happy to answer them.
Thank you very much, Dr. Lane. That was very, very interesting. I did have one question.
Yes.
When you say that the results go out at least 6 months, are you continuing to test these mice, or are we just at the 6-month mark and that's the latest data that you have?
We're continuing past the 6-month mark. It's, so we have animals now that are not yet at 6 months, but we plan to go greater than 1 year and see how long this recovery is mediated.
So far, however far out you've been able to go, the results are promising.
Yes, we see the sustained clinical recovery.
Lisa.
Thank you. Thank you very much.
I wondered how broad the application of this science would be towards other autoimmune diseases. Mm-hmm. And then every scientist in here will probably be rolling their eyes because you know the answer to this, but I don't. Why do women have such a preponderance towards susceptibility?
I'll answer the first question first. So yes, I think that numerous autoimmune diseases could benefit from this type of therapy. And so, for example, diabetes, arthritis perhaps, and that's why we're very interested in finding out what's being secreted by these cells, and does it have an ability to dampen immune cell activation in autoimmune diseases that occur in the periphery. And so this is something that we certainly want to explore. With regards to your second question, that's, there's some elegant work actually being done here by Rhonda Voskuil at UCLA that examines, in particular, MS, but why women may be more susceptible to multiple sclerosis, and then you could extrapolate that to other autoimmune diseases. But in brief, they're components of the immune system that aid in host defense often are amplified in women, and that probably occurs at a molecular and hormonal level. And why that happens, again, there are numerous investigators within the field exploring this, and I wish I had a better answer for you, but I just don't. But this is currently being investigated by numerous different investigators.
Dr. Cesare.
Tom, that was terrific. And it's great to see that you continue to do fabulous work even though you left Scripps.
Scripps was the best training environment I could have been in.
So I'm intrigued, puzzled, worried, and excited by the pleiotropic nature of the effects that you're seeing. And I'm wondering whether I could get rid of all that anxiety if you would tell me that the virus disappears. The fact that the effect is so prolonged and the stem cells have a very short half-life after transplantation suggests that you're getting, or that those cells are getting directly at the cause of the demyelination, the initiating cause, which is the virus that you've infected the mice with. So do you have information about that?
I do. And so what Dr. Cesari is asking is, if you recall, we initiate this disease by infecting the animals with a neurotropic virus, and that's really causing the infiltration of these T cells. And so a very important question, and I'll expand on your question, is A, are the animals recovering, and is this recovery sustained because we've gotten rid of the virus? So in other words, are the neural progenitor cells mediating some antiviral effect that we don't know of? No, they're not. I can tell you that at least at 6 months post-transplant, we can detect viral RNA. Not replication-competent virus, but we never do see replication-competent virus past 3 weeks post-infection. But we're not seeing any swing in viral RNA levels. So no, that's not why those animals are recovering.
Viral proteins.
We've not looked at that. We have not done precise immunocytochemical analysis to look at that. But what I can also tell you, and I think this is very important, is we do not see viral recrudescence. So in other words, the fact that we're dampening neuroinflammation in these animals, remember the immune system is really critical in controlling replication of microbial pathogens, and all of us have persistent viral infections within our central nervous system. And if you dampen immune surveillance, you allow yourself perhaps to have the reemergence of viruses within the CNS, and that can be very problematic. It actually can cause a disease called, for example, progressive multifocal leukoencephalopathy, which has occurred in patients of MS treated with a drug. But we don't see viral recrudescence in these animals. I don't think I alleviated your anxiety, did I, Frank?
That's okay.
Okay.
Dean Pomeroy.
That was just an amazing presentation, by the way.
Thank you.
That one of the best explanations of multiple sclerosis I've ever heard, and really, really exciting results.
Thank you.
So my question is, how much of this, of your positive benefit could be reproduced if you could manipulate the cytokines directly and not have to use the stem cells as the delivery mechanism?
Right. Those types of manipulations are being investigated in different avenues and different laboratories, right, in terms of trying to dampen down disease progression. And so that, again, it goes back to trying to identify what factor or factors in the supernatants of these cells are doing that. And we think we have, I'm very cautious to talk about these data, and so we're interrogating that in more detail, but it does certainly seem to be, if we can identify that, then you could potentially have a single injection or once every 6 months that modulates or dampens these autoaggressive T cells and allows for recovery to occur. But we're looking into that right now very aggressively.
Well, I look forward to the next presentation after you.
Well, thank you. That's very kind of you.
Well, Dr. Lane, again, most interesting. Thank you very much.
Thank you
and
very
keep
much.
up the good work.
Thank you. I appreciate this opportunity.